Okay, so the next topic, all right, and it is also the last topic here. It is a range of a range of reproductive structures or range of reproduction in Bacillareophyceae, that is in diatoms. Now, coming to the reproduction of diatoms, they usually reproduces both by vegetative and sexual means. So there is no asexual reproduction, okay, by vegetative and sexual means. Now the first one here, the vegetative reproduction, it is usually performed with the help of cell division, and it usually takes place at midnight or in the early morning. So to understand the vegetative reproduction, let us take a look at this diagram here. Just see here this one, you can see it is a single unicellular cell of diatom, all right, known as the frustule, remember. So in this frustule, what will happen during cell division? These two theca, that is the hypo and the epitheca. Remember the bigger one, the slightly bigger one is epitheca, and the lower one is the hypotheca. So both these theca, they will start to separate. The protoplast inside it will become enlarges in size, and the cell thus it will increase in volume and slightly separates both the theca. So when they separate, all right, during separation, what happened? Even the protoplast will get separated into half longitudinally, all right, this way. So it will get separated into half. So suppose this is the hypotheca and this is the epitheca, they get separated. So the protoplast also here also it gets separated in the hypotheca and the epitheca, all right. So what happened when these two get separated? The protoplast it becomes naked from this side, all right, it becomes naked because there is no covering right now, because these two theca they have separated. So the protoplast it becomes naked. So what happened, you see in the second diagram here, when these theca they get separated, later on they will behave as the epitheca. Just remember, during the first cell division, both the theca when they get separated, now they behave as epitheca always, all right, they behave as epitheca of the mother cell. So that is why you can see this one black in color. So both here in the second diagram you can see that it is shown in black color. Why? Because they behave as the epitheca of the mother cell. So later on you can see the epitheca will, the silicaceous material will get deposited here, okay, and the epitheca will form in both the daughter cell, all right. So now the division is completed. So next again same thing will happen. So this will again undergo cell division, giving rise to again two daughter cells, you can see. Now what happened here, I will show you. New siliceous valves are deposited, you can see. They are deposited towards the naked side of the protoplast, because this epitheca at first it becomes naked because they get separated like this. So they become, the protoplast become naked. So a new siliceous valves will get deposited here, all right, towards the naked side of the protoplast in both these daughter cells. And also connecting bands are developed between these theca, and later on these daughter cells also they will get separated in the same manner as the first division. So just remember that during cell division, both the theca, that is this the epi and the hypotheca of the mother cell, in the second division just now I had shown to you that they behave as the epitheca of the daughter cells. That is why it is shown here in black color both. You can see in the daughter cells that both these epitheca and the hypotheca, they become the epitheca of the daughter cells, and then only this new wall, siliceous wall, is deposited in the naked side of the protoplast. All right, so I hope till there it is clear. And so at the site where the hypotheca behaves as epitheca, the cell becomes reduced in size. And thus with continuous cell division, some cells, they gradually become reduced in size. So you can see in this diagram, as cell division it continues. All right, as cell division it continues, since the hypotheca, you can see here the epitheca, it is always it is slightly larger than the hypotheca, right? So here in the daughter cell, when the hypotheca it acts as the epitheca, since it is slightly smaller, so this new one which is deposited here, this new cell, it has to be slightly smaller. Again in the next division, since this smaller cell will behave as the epitheca again, so the new deposited cell will be slightly smaller. So as cell division continues, the cell also it becomes smaller and smaller and smaller in size. All right, so this is how cell division takes place in diatoms. So now we are going to study the next type of reproduction, that is sexual reproduction. Now sexual reproduction, girls, if you remember, you have studied classification of the algae, right, with miss. So the diatoms, we have two groups, we have the perennials and the centrales. We have two orders. In the class Bacillareophyceae, we have two orders, that is the perennials and the centrales. So now we are going to study sexual reproduction in both these orders. Okay, because the pattern, so this reproduction, the reproduction in case of sexual reproduction, why we have to study in both the orders perennials and centrales, because it is different. Okay, so the pattern of sexual reproduction, it differs in both perennials and centrales, and during this process we will see that some sexual spores, that is the oxospores, is formed in both the groups, in both perennials and centrales, and these cells, they become reduced in size and they are able to regain their normal size through the formation of the oxospores. So this oxospore formation in both perennials and centrales we will discuss now. So just remember that it is known as a restorative process rather than multiplication. Why? Because you see, during cell division, the cell becomes reduced in size. So during this sexual reproduction, okay, these cells which become reduced in size, they are able to regain their normal size through the formation of these oxospores. Okay, so when they regain their, since they are able to regain their normal size, it is also known as a restorative process rather than a multiplication. So now we will discuss both the oxospore formation in both the perennials and the centrales in sexual reproduction. So with this first one here, that is the oxospore formation in perennials, we will see this one. Now the oxospore formation, it takes place through gametic union as well as autogamy and parthenogenesis. So let us study one by one. First, gametic union, it means the union of the male and the female gametes. All right. So first one here, if you just take a look at the first point here, that is the production of one oxospores by two conjugating cells. Now in this first type here, in this process, what happened? Two uniting cells all right. Two cells which are will unite, will become very close to each other, and they become covered by a mucilaginous sheath, and thus the diploid nucleus of each cell, it will undergo meiosis. So during meiosis what happen, will have one cell will divide to give rise to four cells, all right. So this nuclei, it will undergo meiosis to form four nuclei, and out of these four nuclei, three nuclei will get degenerated and only one will survive. And this surviving nucleus, it will then behave as a gamete. And later on, what happened? These gamete from both these frustules. Remember that the cell in unicellular cell in diatom is always is known as a frustule. Okay, so these gametes, they will come out from the parent frustule and they will unite together to form a diploid zygote. So to understand that, let us see, just take a look at this diagram. You can see what happened in figure A. Two cells or two frustules, they come close to each other, right? They come close to each other, and as you can see, they get enclosed in this covering, okay, a mucilaginous covering or a mucilaginous sheath. And what happened in figure B, just take a look at the nuclei. Now there are 1, 2, 3 in each cell, there are 4 nuclei. That means this nucleus, single nucleus, it undergoes meiosis to give rise to 4 nuclei. And as you can see here in figure B, in each of these two cells, what you see here, that three nuclei, they become degenerated and only one nucleus survive. All right, so these three, which looks like dots here in figure B, they are the degenerating nuclei, and only one nucleus survive, and this one nucleus, it will behave as a gamete, okay, a male and a female gamete. And these gametes, as you can see in figure C, they will come out from the parent cell or the parent frustule, and they start to unite together. You can see in figure C, they will unite together, and after uniting, they will give rise to a zygote. All right, this lower portion here is the empty frustule. Why it is empty? Because both the nucleus from the frustules, from these two frustules in figure C, you can see they will come out from the frustule and they will unite together to form a zygote. So this zygote is diploid, and this is the mucilage, all right, the mucilage sheath, and these are the empty frustules. So this zygote, after a short period of rest, okay, it will undergo a period of rest, and later on, what do you see in figure F? Just take a look at figure F here. This zygote, it elongates considerably, and here it functions as the oxospore. And later on, the oxospore, it projects out from the parent frustule along with mucilaginous sheath, and it elongates in a plane parallel to the axis of the parent diatom. All right, so you can see it elongate in this way, okay, parallel to this parent diatom. And here what happened later on, the oxospore is enclosed in a pectic membrane. Okay, a pectin, it is a polysaccharide substance. Now this pectic membrane is known as the perizonium. So just remember that the oxospore, it is enclosed in a perizonium, and the oxospore, it will then develop new frustule, all right, inside the perizonium. Thus, new diatom cell is formed which regain its normal size. So here this oxospore in this figure which you see, it will uh, get enclosed in a pectic membrane known as the perizonium, and inside this pectic membrane it will develop new frustule. Alright, so new cell will form, new diatom cell is formed, and this new diatom cell which is formed, it regains its normal size. So it regains its normal size, and this type of division, okay, this type of reproduction where we see that two cells, all right, one oxospore is formed by two cells, two conjugating cells. So remember the first type here, just remember it is the production, the first one, it is the production of only one oxospore by two conjugating cells. You can see two conjugating cells here, they form one oxospore. So this type can be found in species such as Coccus placentula, then Surirella, Saxonica, etc. Okay, so these are the two species where this type of reproduction can be found. Now, the second one, that is not only one oxospore will form. Here in the second type, so in some diatom species, two oxospores are formed by two conjugating cells. So here two conjugating cells again will unite, but they will give rise to the formation of two oxospores. So here what happened, this is also a very common process. Okay, so it is a very common process, and what happened in this conjugating, in this process, the two cells will become very close to each other. Again, just take a look at figure A. They start to come close, very close to each other. Again, same thing, they will get enclosed in a mucilaginous sheath, and the nucleus of each cell will undergo meiotic division. Alright, they will undergo meiotic division, and again they will form four nuclei. But what happened here, out of four nuclei, only two will degenerate. So just take a look closely at this diagram, at this figure B here, where you can see two nucleus are written here in dot, very small dot, and two nucleus. So these two nucleus which are here, which looks like just small dots, those are nothing but they are degenerating nuclei, and two nuclei survive in both the cells, you can see. All right, so since two nuclei survive, the cytoplasm, it will divide either equally or unequally. So here in this figure B, you can see that the cytoplasm, it divides equally, but sometimes it can divide unequally also, and along with one nucleus they behave as gametes. Alright, so one nucleus, since the cytoplasm also it divides, as you can see clearly in this figure B, so one nucleus will go in one cell and the other nucleus will go to the other cell, and both these nucleus now they behave as gametes. So two gametes are formed in each cell. So since two gametes are formed in each cell, now the pattern of union between the gametes, it varies from species to species. Alright, it depends from one species to the other. It's not the same. Now both the gametes of the cell, they may be active, and they will fuse with the gametes of the other cell. So you can see here in figure C where these two gametes, they start fusing together. Thus two zygotes are produced in a single cell, or out of two, one becomes active and fertilizes with the opposite one, and thus one zygote is produced in each cell. Now what happened here in figure D, after these gametes they fuse together, after a zygote is formed, just take a look at figure E. The zygote, it will start to elongate, and here it will function as the oxospores. So you can see that two oxospores are formed. Now you understand why two oxospores. The only difference is that in case of the first one that we have just discussed now, we see that out of 4 nuclei, only one become function from one cell. So it will fuse with the other cell and it will form one oxospore. But here, since two nucleus are functional from one cell, so these two, they will fuse with the two nuclei from the other cell, and thus two zygotes are produced and two oxospores are formed. And it will happen in the same manner where these oxospores, they will get enclosed in a pectic membrane known as a perizonium. Alright, so you can see here the zygotes elongate and function as oxospore, and the oxospore, it develop a perizonium around themselves, and both of them, they will develop new frustules on the outer side. Alright, that is the inside of the perizonium. Now thus two diatoms of normal size are formed, and this type is found in Cymbella species and Gomphomima parvulum. Alright, so these are two of the examples of diatoms where this type of oxospore formation is found. Okay, so the first one here, that is the remember that the oxospore formation in pennules, the first one, we have two cells, two conjugating cells, they give rise to one oxospores, whereas in the second one, two conjugating cells, they give rise to two oxospores. And in the third one here, what actually happened, only one oxospore is produced by only one cell. All right, so this process of oxospore formation is called pedogamy. So remember girls, this one they used to ask sometimes. Okay, suppose if they ask you in viva, so please remember that when only one oxospore is formed by one cell, this formation is known as pedogamy. And in this process, what actually happened, okay, a single cell, it has a diploid nuclei. Okay, that one nucleus, it will undergo meiosis again to form 4 haploid nuclei. Now what happened, two out of the 4 nuclei, two partially degenerate. All right, two, they will undergo degeneration, and each of the rest two, along with the cytoplasm and one partially degenerated nucleus, behave as gametes. So later on, what happened, the union between the two sister gamete takes place and form the zygote. So here, just remember, out of the 4 nuclei, 2 nucleus will undergo they will partially degenerate. All right, and the other two, okay, while the other 2 nucleus, along with the cytoplasm and one of the degenerated nucleus, they behave as gametes, and these, they will undergo union. Okay, they will undergo union and they will form a zygote. So later on, what happens, suppose this is a single cell, then I have a nucleus. So this nucleus will undergo meiosis and will give rise, all right, will give rise to 4 nuclei. So out of the 4, 2 nucleus are partially degenerating, whereas 2 will behave as gametes and they will fuse together to form a zygote. So in this way, that is why only one oxospore it can be produced by only one cell. So it is same thing will happen, that the zygote, it will come out from the parent frustule and it will behave as an oxospore. Again, this oxospore, it will get covered by a perizonium and develop walls inside the perizonium. So thus one diatom cell of normal size is formed. All right, so it's the process is the same as in case of the first two that we have already studied. So here I did not show any diagram, but just for you to remember that how come only one oxospore is produced by only one cell, it is because In this case, the nuclei, when it undergoes meiosis to form 4 haploid nuclei, 2 nuclei degenerate and 2 nuclei they will function as the gametes, and they will unite to form a zygote. And the fourth one, we have production of one oxospore by autogamy. Now in this process, the diploid nucleus, it undergoes first meiotic division. All right, so when it undergoes meiotic division, 2 haploid nuclei are formed. Now see, when they undergo meiosis, we know that meiosis is a 2 process, right? First, the nucleus will divide. The first meiotic division, what we have, we will get 2 daughter nuclei, and in another division we will get 4 nuclei. That we know, it is a 2-step process. So what happened here in the first process, all right, these 2 haploid nuclei here, the 2 nuclei in the protoplast, they will come side by side, and actually after this they will fuse together. Here meiotic division occur. This nuclei will give rise to 2 daughter nuclei in the first meiotic division, and these 2 haploid nuclei in the protoplast, they will come together, they will fuse again, they will form a 2n nucleus, a diploid nucleus. So this is called autogamous pairing. Okay, it is known as autogamous pairing. And this protoplast, along with the nucleus, diploid nucleus, it comes out from the parent frustule, and directly it behaves as an oxospore. Alright, so instead of undergoing, just now I had shown to you, in meiosis, what happened in the first meiotic division, we get 2 nuclei, and in the second meiotic division we will get 4 nuclei, right? But what happened in this process, actually only the first meiotic division occur. After the meiotic division occur, these nuclei, the second meiotic division does not occur here. So we have only 2 nucleus, and these 2 haploid nuclei, instead of undergoing further division, they will actually fuse together, they will pair together to form, and they will behave as an oxospore. Okay, so when they fuse together, they will give rise to a diploid nucleus. So this diploid nucleus, it will come out from the parent frustule. So this step is not there. It will come out from the parent frustule, and it will behave as oxospore. Again, it will be covered in a perizonium, and this new individual diatom will of normal size will develop. So this type, it can be found in species such as the Amphora normani. Okay, Amphora normani, this type of oxospore formation can be found. Now next type, we have the production of oxospore by parthenogenesis. Now what happened here, the diatom cell, they come together. All right, the first step, the diatom cell, they come together, covered by a common mucilage, envelope, or a sheath or envelope, it's a covering. Okay, they get enclosed in a covering, in a mucilage covering, and the diploid nucleus, it will undergo 2 sequential mitotic divisions. All right, so here in this case there is no meiotic division. You know that in meiotic division, from a single cell we finally get 4 cells, right? At the end of meiotic division, we have 4 daughter nuclei, but in this case meiotic division does not take place. Instead, mitotic division occurs. So we know that after mitotic division occur, 1 nuclei will give rise to 2 daughter nuclei. So you can see in each of these cells in figure B, we have 2 daughter nuclei. Now 1 nucleus here, it will degenerate. So as you can see clearly in this figure that in each of these cells, one nucleus it degenerate and the other one diploid nucleus along with the protoplast it remains inside the cell. Alright, this will actually come out of the mother cell and it will behave as the oxospore. Again, now this oxospore it will then covered by a perizonium and secrete a new wall around itself, and thus normal cell is formed. Okay, so this is a simple division which occur by parthenogenesis. That means in each of the single cell, actually these nucleus you can see here in each cell, they will these you can see in figure B what happened when mitotic division occur. One nucleus will degenerate and the other diploid nucleus along with the protoplast it remains, and this will come out and behave as the oxospore. Okay, so actually these they will not fuse together, just like you have seen in the first case where these two they will fuse together, then they will come out and behave as the oxospore. So here actually we get two oxospores. Why? Because these they will not fuse together, instead they will directly, without fusion, they will directly behave as the oxospore and give rise to a new cell, normal cell. All right, new diatom cell. So that is why here we have two diatom cell. This process is usually known as parthenogenesis. That means the gamete itself is giving rise to a new individual without undergoing fusion. Just remember this, why it is known as parthenogenesis. In figure 1 here, as you can see that just take a look at figure C and D. You can see in figure C, these they will come out, the cells will come out from the frustule, they will fuse together and then they give rise to a single zygote. Then this zygote, as you can see in figure F, it behave as the oxospore. So that is the difference. All right, so in figure 5, why it is known as parthenogenesis? Because these nucleus, the diploid nucleus here, they do not fuse, they do not come out from the parent cell here, they will not come out and they will not fuse, instead each will give rise to a new individual. They will act as the oxospore and will give rise to a new individual without undergoing fusion. That process is known as parthenogenesis. All right. So the last type here in pennials, we have the production of oxospores by oogamy. So this oogamy we have come across many times when we study the life cycle of Ectocarpus, Chara, Edogonium. You have come across this word many times, oogamy. I have mentioned that it is the fusion of the gametes which occur between two unequal gametes. And actually the female gamete is always larger in size, whereas the male gamete is smaller in size. Alright, so that is known as oogamy. So in this process, you can see here that the female, that is the oogonium, alright, it is larger in size. You can see here it is larger in size, the oogonium. And what happened here in oogonium, it will undergo meiosis. So you know that after meiosis we get 4 nuclei. All right, so one nucleus it undergoes meiosis to give rise to 4 daughter nuclei. And also just take a look at this portion here, this one here. Actually the protoplast it also divided into 2 unequal parts. You can see this cell here is quite smaller and this is larger. So the protoplast also is dividing into 2 unequal parts, each containing 2 nuclei. Just take a look closely at this diagram here. You can see this is the first nucleus, this is the second nucleus, which is quite small. Here also we have 1, 2. So the lower half of the cell of the oogonium you can see is larger, and this it behaves as a functional ovum. Alright, and the upper one is smaller and it is a non-functional ovum. This one, it is a non-functional ovum, abortive ovum. And now the functional ovum, okay, it contains one functional nucleus and one, this you can see the nucleus here which is bigger in size, it is the functional nucleus, whereas this one which is smaller in size, it is non-functional. And at maturity it will starts to degenerate. So just remember that the oogonium here, it will divide it into two on equal half, the lower one which is slightly bigger and the upper one which is smaller. So the upper one, it is the non-functional ovum, that is the abortive ovum, whereas the lower one, it is the functional ovum, and both these they contain two nucleus. The degenerating nucleus, you can see the smaller one, whereas this larger one, it is the egg nucleus, that is the functional nucleus. And later on, what happened here, the male cell, you can see the antheridium in figure B here. This antheridium also it undergoes meiosis. Again also it also form four nuclei, and the protoplast here in the antheridium also you can see it divides into two parts, thus two microgametes are formed. Okay, and each of these gametes they contain two nuclei each, you can see here. In antheridium you can see it has divided into two cells, and each of these cells they contain two nuclei, of which one is functional and the other, which is written in a, this smaller one is non-functional, the bigger one is functional. All right, so the microgametes, they are naked, they are globular, and they are also non-flagellate. Non-flagellate meaning they cannot swim, they don't have any flagella. Now after coming out, these male gamete they fertilizes the egg and form the zygote. So you can see here in oogonium, these microgamete, you can see they will come here. All right, these microgamete they will come in contact with the female gamete and they will fertilize the female gamete to form the zygote, and later this zygote will function as the oxospore and will form a new individual of normal size. So this type of division, it is known as the oogamy. Why? Because it is production of oxospore is by oogamy. Oogamy meaning the fusion of the male and the female gamete, where the male is larger or the female gamete is larger in size as compared to the male gametes, that is antheridium. All right, and it is found in this type of species, that is Rhabdonema adriaticum. So this is one of the species where this type of sexual reproduction is seen. So now coming to the last part, the next part, that is, so you see that there are 6 type of oxospore formation in pennials. Remember there are 6 types. All right, I will not repeat that. If you want to remember, you can just go and just watch again the video to remember what are these 6 types of reproduction, sexual reproduction in pennials. Now coming to the second type of order in Bacillareophyceae, that is centrials. Here it is quite simple as compared to the pennials. It takes place only by 2 types, that is autogamy and oogamy. So what happened in oxospore formation by autogamy? It is simple. You can see that the protoplast of the vegetative cell, you can see here in the first figure we have a frustule. All right, a frustule or a cell. Now it will secrete a mucilage. All right, it will secrete a mucilage, and both the theca will get slightly separated, and the nucleus will undergo, you can see this round here, the round portion here, that is the mucilage, okay, which enclosed the nucleus, and thus these theca, they are slightly separated, and the nucleus undergo meiosis, forming four nuclei. So out of the four nuclei, two nuclei will degenerate, and other two, they will fuse together to form a zygote. So this zygote, it will form after resting, after a period of resting, it will form it will act as the oxospore, and this oxospore, it will develop into a new frustule or a new cell. It is enclosed inside a perizonium covering, and it will form a cell of normal size. Right, so it is simple here. This process, this is called autogamy. All right, where the nucleus of a single frustule, it undergo meiosis to form four nuclei. Two nuclei will degenerate, and two nuclei will fuse together to form a zygote, and this zygote will finally form the will act as a oxospore, get enclosed inside a perizonium, and it will develop into a new individual cell. Okay, that is new frustule. Now the next second type is biogamy. So oogamy is similar, just like in case of here in the, you see in oogamy, in just now we have studied in pennials, we have it takes place by the fusion of the egg and the sperm develop inside the oogonium and the antheridium respectively. All right, so here we have oogonium and antheridium. So in oogonium, the egg will form, in the antheridium the sperm will form. So here in oogonium, a single vegetative cell, it will behave as oogonium. The protoplast of oogonium will undergo meiotic division, and it will form four nuclei. Right, out of the four, three will degenerate, and only one one nucleus will function as the egg. While in case of antheridium, the pattern of development of sperms varies in different species. So in some species like Melocera here, okay, variants of protoplast undergo meiotic division, and they will form four haploid nuclei, and each haploid nuclei with some protoplast, it will develop into into a uniflagellate sperm. Okay, uniflagellate meaning having only a single flagella. This is referred to as tinsel type. Tinsel, just remember tinsel type. That means those which has only a single flagella. And in while in other species, here it is the case of Melocera. All right, suppose in other species of diatoms, sometimes the number of sperms may go from 8 to even 128 in number. So now you understand the formation of the egg and the sperm. So what happened when these two, they form? After coming out from the antheridium, you can see only one sperm, it will enter inside the oogonium, and it will fertilize with the egg. All right, so in this figure, what do we see? After these four sperms are formed here, and even here you see that three in oogonium. In case of antheridium, four sperms develop, since this nucleus will undergo meiosis to form four nuclei, and these will develop into sperms. Whereas in oogonium, we have will again undergo meiosis, but three nuclei are degenerating. Only one nucleus will act as the egg. All right, the functional egg. And here this sperm will, you can see the sperm from here, it will fuse with the egg, and after they fuse together, they will give rise to a zygote. All right, so you can see in fertilization, this zygote, you can see the resultant zygote, it will then undergo mitotic division, but one nucleus In mitotic division, you know that from this zygote, all right, the zygote which is diploid, it will undergo mitotic division uh, to give rise to haploid nuclei, and these the remaining one of the nucleus, it will degenerate, all right, it will degenerate, and this remaining nucleus, it will behave as an oxospore. Then the oxospore, it will develop new wall inside the perizonium covering, and it will form a new cell of normal size like the mother, which is also known as the firstling cell, right, it is known as the firstling cell. And thus, from the above process of sexual reproduction in both the pennials and the centrials, it becomes clear that sexual process in diatoms does not lead to multiplication but to regain its normal size. All right, you can clearly see from all these processes the sexual reproduction in diatoms, it does not lead to multiplication, the cells does not multiply, but you can see it's just that the cell, it regains its normal size. That is why uh, you can see here, remember in cell division, sorry, in this one, the first one here, let me show you. Okay, where is that? Okay, I have already shown that time. Just remember, because you can see clearly from the sexual reproduction that it is a regaining of normal size rather than multiplication. Whereas in vegetative reproduction, you can see the cell division occur. All right, so multiplication occur, but in case of sexual reproduction, the sexual process, it actually does not lead to multiplication of the species, but instead in order it is only to regain its normal size. And also we have just want to show you this one, actually that is the end of the range of reproduction. All right, so this just to show you what are the resting spores. These are the spores which form during unfavorable conditions. Suppose the climatic condition is not favorable for the growth of the diatom, then these type of spores will form, known as the resting spores. So some members, they reproduce by the formation of a thick walled resting spores known as the cyst or the statospores. Okay, and they are formed in this species Melocera. So in case the environmental condition, suppose the pH of the water or the temperature or anything, or the climatic condition is not favorable for sexual reproduction to occur, for oxospore formation, then instead of oxospore, these resting spores known as the cysts or the statospores, they will form, and that later on, when the condition is favorable, then they will undergo sexual reproduction. All right, so these are the range of reproduction in both the pennials and centrials in diatom.